after getting in-depth knowledge about types, production, and recruitment of phagocytes. Let's move forward to action of phagocytes, that is phagocytosis. Usually all phagocytes are equipped with some intracellular compartments like lysosome that carry microbicidal enzymes like lysozyme, nucleases and proteases, lipases, and substances like hydrogen peroxide. Apart from some variations in their content of lysosomal enzymes, all these phagocytic cells use essentially similar mechanisms to phagocytose foreign objects. This mechanism of phagocytosis consists of series of events including attachment, endocytosis or ingestion, and digestion of microbe. Starting with the first step, that is attachment. Following chemotactic signals, phagocytes like neutrophils reach at the site of infection. Similarly, other phagocytes are also guided by chemotaxis. Let's look at its proper definition. That is the unidirectional migration of cells along a concentration gradient of a chemoattractant. And that chemoattractants are usually inflammatory cytokines or microbial products. So, following chemotaxis, once phagocytes reach to the site of infection, now they are in close proximity with microbe. Here the process of phagocytosis activates, and this activation is triggered by binding through one of the receptors present on its surface that we have already discussed. We know very well that phagocytes contains pattern recognition receptors, PRRs, receptors for antibodies, that is Fc receptor, and receptor for complement binding, that is C receptor. In general, PRR on phagocytes can detect pathogen-associated molecular pattern, PAMPs. These are actually a diverse set of microbial molecules that share a number of different general patterns or structures that alert immune cells to destroy intruding pathogens. PRR can bind to molecules that are shared among infectious microbes, but are not found in vertebrates. Normally, the most common pathogen-associated molecular patterns or PAMPs are usually mannose-binding lectin, NBL, that are the complex carbohydrates found in the cell wall of yeast, and lipopolysaccharides, LPS, that is cell wall component of gram-negative bacteria. This pattern recognition of pathogen by soluble or cell-associated PRR can lead to direct lysis of the pathogen, enhancement of phagocytic cell functions, production of antimicrobial proteins, and production of cytokines and chemokines. This mechanism of attachment between PRR and PAMPs is common for non-capsulated pathogens, but the capsulated ones can't be detected by PRRs. So, for their attachment with phagocytes, they need another additional step that is called opsonization. Greek opson means to prepare for eating. So basically, opsonization is the process by which bacteria and other cells are made available for phagocytosis. For this purpose, they get coated with some protein or molecule, and the molecules or proteins that help to bind pathogens to phagocytes and stimulate phagocytosis are known as opsonins. Usually, C3 that is produced during complement activation, and antibodies mostly IgG can act as opsonin. C3 is the most important opsonin and binds to 3 different receptors present on a range of phagocytes. During alternative complement pathway, C3 is split by convertase enzyme, and its cleavage product that is C3b is formed. C3b is very common and important opsonin. It gets covalently linked with the microorganism and acts as an opsonin. In this way, complement aids in opsonization, and thus in phagocytosis. Other than complement, antibody also opsonizes microbes for phagocytosis, both directly by Fc receptors and indirectly by complement activation, as cell walls of invading pathogen and of phagocytes are negatively charged and hence they repel each other, but opsonization or coating of microbe basically work by overcoming this repellent force between cell walls of microbes and phagocytes and promote uptake of the pathogen by the macrophage. In this way, by attachment of PRR with PAMPs or by following opsonization with either complement or antibodies, pathogen gets recognized and then attached by the phagocytes. After attachment, next step in phagocytosis is endocytosis or ingestion. As we discussed at the beginning, that endocytosis is the process to take in substances from outside the cell. Same is needed for phagocytosis now. When the foreign particle or pathogen gets attached at the receptors of phagocytes, either directly on PRR or on Fc receptor or C receptor, it is then pinched inside of the cell, together with a part of phagocytic cell membrane. This vacuole, that is composed of phagocytic cell membrane with a captured pathogen inside, is called phagosome. Let's look at its proper definition. That is, a phagosome is a vesicle, a small cellular structure filled with fluid and cellular waste. Its goal is to destroy whatever is trapped inside it, such as a pathogen or cellular debris. We know that all phagocytic cells contain intracellular compartments or vesicles. One important vesicle that contain antimicrobial substances inside are lysosomes. So, once the particle is internalized inside the phagosome, this vacuole can fuse with lysosomes to form another vacuole called phagolysosome. This phagolysosome is a microbicidal vacuole, and it contains many substances that have ability to kill microbe. This formation of phagolysosome is also called as vacuole formation. Within these microbicidal vacuoles, invading particles are further processed. Now we move towards final step of phagocytosis, that is killing or digestion of microbes. A lysosome or phagolysosome is able to break down the stuff inside of itself by three mechanisms. Firstly, it drastically lowers the pH of its internal environment. Lowering the pH makes the environment inside the phagolysosome, makes it very acidic. This is an effective way of killing or neutralizing whatever is inside the phagolysosome, so it cannot infect other cells. Also, phagolysosome, that is a microbicidal vacuole, contain a number of hydrolytic enzymes or digestive enzymes, including cathepsins that are the most abundant lysosomal proteases and play role in intracellular protein degradation. Proteases that catalyze the hydrolysis of microbial proteins. Lysozymes that functions as an antimicrobial agent by cleaving the peptidoglycan component of bacterial cell walls. And lipases that catalyzes lipid destruction. While other microbicidal components of the phagolysosome are scavenger molecules, such as lactoferrin, that sequesters the iron required by some bacteria, ascorbate, that interacts with copper ions and hydrogen peroxide and can be bactericidal. Along with that, this intracellular killing of many bacteria requires the uptake of oxygen by the phagocytic cell. So all these events initiates respiratory and oxidative burst. Oxygen that enters from outside the phagocyte can be further processed by NADPH oxidase that generates superoxide, O2 negative. These superoxide can then dismutate to hydrogen peroxide, H2O2, by superoxide dismutase, which can in turn react with O2 negative to generate more complex reactive oxygen species, or ROS, such as hydroxyl radicals and singlet oxygen. ROS are highly toxic to many microorganisms, but excessive ROS production may contribute to damage to host tissues. So, these reactive oxygen species are rapidly removed by cellular enzymes such as catalase and glutathione peroxidase. In addition, hydrogen peroxide can be combined with chloride ions into hypochlorous acid by the enzyme myeloperoxidase that cause microbial destruction. Reaction of antigens with hypochlorous acid may also enhance their recognition by T lymphocytes. So all these things like acidic medium or low pH, digestive enzymes, and oxidative burst cause killing or digestion of trapped microbe, leaving behind a sac filled with indigestible material called as residual body. So, once the contents have been neutralized, the residual body then travels towards the boundary of the cell and is eventually discharged from the cell by a process called exocytosis. Here, phagocytes can also undergo partial digestion of microbial protein that can be displayed on the surface of the APC associated with an antigen-presenting molecule, such as MHC class I or MHC class II, for recognition by T and B lymphocytes. Such process is called antigen presentation. In this way, they present antigen to more specific cells of adaptive immune response, that are T and B lymphocytes, for further processing. So this was all about detailed mechanism of phagocytosis. Let's summarize it briefly. Phagocytosis mainly consists of three main steps, that are attachment, endocytosis, and digestion or killing. In attachment, first recruitment of phagocytes and recognition of pathogen is aided by the process of chemotaxis. Then these pathogen gets attached at particular receptor site and activate phagocytes. This attachment initiates process of endocytosis, where first pathogen gets pinched in together with phagocytic cell membrane, making a vesicle called phagosome. This phagosome then gets fused with internal cellular compartments that are lysosomes. This gives rise to another vacuole called phagolysozyme. This is main microbicidal vacuole. After this, endocytosis and vacuole formation inside the cell. Process of digestion or killing of microbes starts. This process gets aided by low-H digestive enzymes and other scavenger enzymes inside the vacuole. Here either it will get completely killed, leaving residual bodies behind, or it will partially digest the microbe and present over its surface as antigen, which is then presented to adaptive immune lymphocytes for further processing. So this was all about it. This video must have improved your knowledge. For more such videos, keep watching sqadia.com.